In Chapter 5, Section 2, we are going to look at active transport, which is moving molecules across a cell membrane against a concentration gradient, or up a concentration gradient, which is different from what we've been talking about before, which was passive transport, and with that form of transport, we're going down the concentration gradient. Now, with active transport, energy is going to be required. We're trying to move particles from low concentration to high concentration, and that needs energy in order to occur. One of the most common forms of active transport in the cell is called the sodium-potassium pump. Generally, when you see the word pump, that should indicate that it is a form of active transport and will require energy in order to function. The sodium-potassium pump is a system of proteins, carrier proteins, that help to transport potassium or K plus ions into the cell and sodium or Na plus ions out of the cell. It's important that we keep the levels of potassium and sodium to what they should be in the cell in order to help the conduction of our electrical impulses in our nerve cells. So to make our nerve cells function quickly and efficiently, we need to make sure that the potassium and sodium levels are regulated properly, and that sodium-potassium pump is what helps us do that. Another form of active transport is endocytosis. And with endocytosis, the cell is going to surround and engulf substances and bring them in. So these are generally either groups of molecules or just large particles that don't fit well through carrier proteins. There's two types. If you're referring to bringing fluids or solutes, so solids that have been dissolved in liquid, that type of endocytosis is called pinocytosis. And if you're talking about large solid particles such as a bacterium, then that would be phagocytosis. You can see in the diagram here, the cell membrane actually forms this little well, okay, or in this one here, a little well where the particles can come in, and then the membrane will start to pinch off until it is completely sealed from the outside and forms a vesicle that will bring those materials into the cell. Now, the exact opposite is called exocytosis. So instead of bringing materials into the cell, this time we are trying to release or exit molecules from the cell. So here we start with a vesicle that might be full of waste or toxins, or could just be proteins that are needing to be sent somewhere else in the body. And that vesicle will come up to the cell membrane, fuse with it, and then open up to allow those materials to be transferred out without exposing the rest of the inside of the cell to the outside conditions.